Hello everyone, my name is Samuel Lashof-Regas and I'm from UCSF in the lab of Leo Segru. The title of my talk is Cortical and White Matter Microstructure of Dimensional OCD Symptoms in ABCD. I have no relevant financial disclosures. So what I'll try and convince you of today is that by leveraging a large, homogeneously imaged cohort and utilizing an approach to imaging analysis that incorporates dimensional, categorical, and longitudinal psychiatric data, we can begin to uncover structural brain correlates of OCD, and that this approach can potentially be applied to other psychiatric conditions in large neuroimaging datasets. It's well known that, and much discussed, that imaging tests for psychiatric disease have been frustratingly elusive. MRI studies are expensive and often have small sample sizes, which poses challenges to internal and external validity, and psychiatric disorders are notoriously heterogeneous. Some groups have admirably tried to solve the issue of small sample sizes by pooling data across sites. However, these efforts have been hampered by pooling heterogeneously imaged patients, which have resulted in associations that don't align with the rest of the field. Here you can see data from one of the most prominent consortia studies, the Enigma group, showing decreases in cortical thickness in both pediatric and adult patients with OCD compared to healthy controls, mostly confined to the occipital region, a region typically associated with visual processing. For our analysis, we looked at data from the Adolescent Brain and Cognitive Development Study, or ABCD, which is a 21-site study of around 12,000 kids who were 8 to 10 years old when they were recruited and are receiving comprehensive, harmonized, multimodal imaging and health data collection every 2 years for at least 10 years. In order to overcome some aspects that might be obscuring effects in past studies, we did a couple things differently when figuring out how to analyze the ABCD data. One of the things we did was to take a hybrid approach to defining how we were going to look at OCD. First, we took all the children who were diagnosed with OCD based on DSM criteria. Then, within that group, we looked at dimensional OCD symptoms based on the Child Behavior Checklist, or CBCL, which is a well-validated tool to assess psychological symptoms. What I'm showing you here is a correlation matrix of the CBCL questions having to do with OCD among children who met criteria for DSM diagnosis of OCD. What we notice is that there seem to be two behavioral phenotypes, one having to do with anxiety-related symptoms in the top left, and one having to do with more classic OCD symptoms of obsessions and compulsions in the bottom right. What this is telling us is that the scores on the anxiety questions all tend to go together, and scores on the obsessions and compulsions questions also tend to go together. While in previous studies, these symptom clusters would have been averaged together under either the DSM diagnosis or the CBCL aggregate score, our approach allows us to analyze the distinct representation of each of these axes in the brain. To analyze brain volumes, we are using an algorithm developed by Anders Dale and colleagues at UCSD called Fast and Efficient Mixed Effects Analysis, which allows for the application of mixed effects models to vertex-wise imaging data, and is able to account for fixed and random covariates in a computationally accessible way. Doing so, we see that these two symptom clusters do seem to be associated with distinct patterns of cortical surface area. The brain map on the left is from a model that took in all children diagnosed with OCD and analyzed cortical surface area associations with the anxiety-related subscore, while the map on the right represents a similar model, but the independent variable was the obsessions and compulsions subscore. For obsessions and compulsions, we can see that there is some change in the cingulate and frontal areas. The final piece of our approach was informed by the fact that while many children will exhibit obsessive compulsive-like behaviors, even to the point of meeting DSM criteria for OCD, not all, or even most, will go on to continue meeting criteria in later childhood or adolescence. In ABCD, of the initial around 900 or so children, 8 to 10 years old, who met criteria for OCD, only around 200 will go on to maintain the diagnosis 2 years later. This fact made us wonder if there was something neurobiologically different about the kids who continued to meet OCD criteria than those who didn't. Here we can see that when we model the effect of obsessions and compulsions subscore on cortical area, filtering for only kids who persisted with OCD over time, the effect is much more pronounced, becoming more obviously bilateral and increasing in effect size. So, what do these structural changes have to do with behavior or treatment? To answer this question, I'll take a slight detour to briefly discuss some additional background. First, I'd like to highlight the overlap between the cortical areas that stand out in our analysis and cortical areas that have been consistently found to participate in fMRI studies, in what has become known as the salience network. As many of you will know, the salience network is a group of functional brain regions that act together and are thought to be involved in response selection and response inhibition, and has been implicated in various forms of psychopathology, including OCD. As you can see from this review from Sarah Peters and colleagues in Toronto, the areas that pop out in our analysis correspond to the cortical nodes of the salience network. Furthermore, I'll mention this paper put out by Andreas Horn and colleagues who analyzed patient responses versus non-response to deep brain stimulation for OCD and found that the discriminating factor was which white matter tracts were being stimulated by the device. They found that the anatomic course of fibers that discriminated between responders and non-responders connected structures in the basal ganglia to the dorsal anterior cingulate and the ventrolateral prefrontal cortex. So just to be clear, the cortical areas that, in our analysis, are associated with severity of obsessions and compulsions map on well to areas that are associated with clinically important white matter tracts and are well supported in the fMRI literature as participating in the salience network. These data led us to ask, using the automated fiber quantification package developed by Jason Yeatman and colleagues at University of Washington, is what do we find when we look at the white matter tracts directly? Indeed, we see effects on certain white matter tracts related to these areas that are specific to the persistent OCD group. Here, I'll ask you just to pay attention to the red lines, which represent the group that continue to meet OCD criteria over time. And we can see that the tracts that were found to be associated with treatment response, such as the anterior thalamic radiation, the cingulum bundle, and orbitofrontal corpus callosum, have altered FA values in the persistent OCD group versus the rest of the groups. Also worth noting is that these changes seem to be specific to these tracts. If we look at the corticospinal tract and the occipital corpus callosum as controls, the persistent OCD group looks basically the same as the other groups. And this brings us full circle back to our objective. Hopefully I've been able to show you that by leveraging a large homogeneously imaged data set and taking an approach to psychopathology that incorporates categorical, dimensional, and longitudinal data, we can start to gain some traction on the problem of structural correlates of OCD that has been vexing the field for a while now. In conclusion, I'll review that changes in cortical surface area in the anterior cingulate and dorsolateral prefrontal cortex are associated with dimensional obsessions and compulsion symptoms in a group that continue to meet criteria for OCD over time. These areas have previously been linked with the salience network, which is hypothesized to play a central role in OCD pathophysiology. These cortical areas are also associated with white matter tracts that predict clinical response to DBS for OCD and were found in our analysis to show FA differences in the persistent OCD group. In terms of future directions, we hope to leverage the same approach to explore other data modalities and other psychiatric disorders. In conclusion, I'd like to thank my mentor, Leo Segru, for all of his support, Andreas Rauschecker, Pierre Nedelec, and the rest of the RSL lab. And I'd also like to thank the OHBM conference organizers for inviting me to speak. Thank you very much.